All right. I think people will keep coming in. I think in the interest of time, we should probably go ahead and get started. So we have around 70 participants right now. So that's great. So again, it's my great pleasure to introduce Dr. Ying Xu, who's our IOB seminar speaker today. And for those of us who are in bioinformatics, Dr. Xu needs no introduction. He's the eminent scholar of bioinformatics and chair of computational biology and professor of biochemistry and molecular biology. He's also the founding director of the Institute of Bioinformatics for the new students, IOB students who are just joining our program. Dr. Xu has done pioneering work in the area of bioinformatics. He's published over 300 papers broadly covering microbial genomics and structural bioinformatics. And more recently, he's really focused his effort on in the area of cancer systems biology, really doing deep data mining and really trying to understand the evolution of cancer, which is a fundamental question in the area of cancer biology. So, so without further ado, I want to just pass on the mic to Ying. So Ying, you have about 75 students in this, participants, and they're still pouring in. So hopefully, you can get started and really tell us a little bit about the work that you're doing in your lab.
Okay.
Take it away.
Thank you.
Yeah. Mm-hmm.
Thank you so much, Kenan, for the introduction. What I'm going to tell you is a data mining story about cancer. So the question we ask is why cancer cells continuously divide, even though the cost for doing so are very high. So we'll get to that point why the cost is very high. So because usually we would think that cancer evolves for some reason, but the cost, everything seems to be against them, then why they are still doing it. So we study public data. We mainly focused on genomic and transcriptomic data, mostly from TCGA database and the GEO database and some other databases, mainly for normal tissues, because we have to do quite a bit of comparisons. And so basically the one probably a little difference compared to other people's work, we focused on basic balances at the chemistry or physics level, things like balance of pH, redox balance, reduction and oxidation balance, and even cell population balances of different types. Then also some physics, like electrical neutrality. So we ask questions at that level. So then we rigorously follow the evolutionary principle of molecular biology. So this is kind of the overline of the presentation. So before I give my seminar, I will present about 6 or 7 slides, giving some background information. I called it a primer. So basically, we'll focus on, for this particular talk, we'll be focused on cytosolic pH. So we know that this is not our work. So for this part, I focused on the primer, and mostly the work is not done by us. Mostly it's done by other people who already have these things established. So we know that the intracellular pH for stationary cells that do not divide, is about 6.8, 6.9. It's slightly acidic. But then for any dividing cells, including both cancer and the normal cells, the pH is 7.2 to 7.4. So you think this seems to be a small change, but this change is very profound. The reason cells, virtually for all cells, for bacteria cells, for human cells, for I'm not sure about plants, but for all animal cells, I have studied, I have looked at the papers that they all have 2 states, pH states. One is for stationary states, one is for the dividing states. I guess evolution just worked out that way to use two states as a regulator. So basically, when cells divide, they use certain type of machinery. When cells doing normal functions, they do other set of functions. So different pH it's a very good regulator. So for people who have done any work related to protein folding, you may know that pH is fundamentally important, because when the pH changes, protein will fold into different shape, because pH essentially changes the proton concentration of the solution. So that will directly impact the hydrogen bonding. So you can imagine that's very, very important. So there are some diseases that's related to the change of pH, which leads to misfolding of protein. For example, I also study a little bit of Alzheimer, which is related to that pH issue. So it is very, very important to keep the stability of the pH for both states. Okay, so because of the importance of cells, human cells and other cells, they all have a pH buffer essentially to keep the pH stability, even when there is any fluctuation. If some reactions may produce protons or may absorb protons, it may affect the local pH, but the system have a buffer to absorb pH, absorb a proton when the pH is too low, and it will release a proton when the pH rises up too high. So then the one thing we know is that this pH has a certain capacity. So if you produce too much protons or too little or too much hydroxides, so at that point, the pH will change. So that will lead to something called acetosis or alkalosis. Okay, but then when you think about this, you may be knowledgeable about cells all have proton importers or exporters. Then you may ask, what if the pH is too low, too high? Can we use the proton importers, exporters to keep the stability of the pH? The answer is yes, but only for a short period of time, because there's a fundamental reason that remember, protons are positively charged. So if you keep importing protons into the cell or exporting protons out of the cell, you will change the neutrality of the cell, which is absolutely big no-no. You cannot change so cell always needs to be in the neutral state. So once you change that, the cell will die. So basically, proton importers, exporters, they can serve as a temporary solution. So that's kissing. There are some sophisticated ways to get around that, but it's not sustainable. So we'll not get into further details about this. Okay, so another part of the primer is about chronic inflammation. So people have, researchers in the field have generally accepted the idea that chronic inflammation is causally linked to cancer. But the detailed mechanism remains not quite clearly figured out. So we'll talk about this. We'll present our view about the immune system and the cancer. So one thing we are going to use is when we talk about chronic inflammation, it has these immune cells will do something called the respiratory burst. They just continuously put out hydrogen peroxide, H2O2, or superoxide. Is this a free radical? And these things are intended to kill invaders, but when you keep doing that, the tissue will be damaged. So this is has been long recognized that it's a very important piece of the puzzle of cancer development. Okay, so that I do want to make one point here, because I feel like in some of the literature there seems to be quite a bit of confusion from my perspective, that actually in the cancer environment, majority of the immune cells, whether it's innate immune cell or adaptive immune cells, they're there to do tissue repair, not to killing non-self cells. So I just want to make sure people, at least for this talk, they're there mainly to do tissue repair, including T cell. So that's why some people consider asking the question why clearly they see T cell in the cancer environment, but they do not seem to do their work, because these cells, if you're looking deep into this, they are there to do tissue repair or to do coordination of tissue repair. Okay, so that's all I'll say for this talk. Okay, so another important piece of our framework is that iron plays a very important role. So we know, based on our literature review, that all cancer tissue has iron overload, based on based on the information we collected. All the cancers in TCGA, 30-some cancer types, they all have some people already noticed each one of them have local iron overload. So when iron meets with chronic inflammation, something bad will happen, something that it will have a substantial change in terms of the level of damages. Okay, in terms of the iron, the relationship between iron and the cancer, there have been quite a bit of epidemiology studies that establish the strong connection between the two. For example, that this is in a plot made by some Japanese researchers. They found that the national beef consumption rate per capita strongly correlate with colon occurrence in that country. So for example, this is the top one is New Zealand. The second one is the US. So you can see that there is a very strong correlation between beef consumption per capita and colon cancer. And also actually there are other cancers, there are similar data. So then also in terms of iron and iron relationship between for iron and cancer, I think there are lots of other data pointing to that direction. So for example, we know that once iron gets inside us, they do not leave, only if you lose blood. So that's the reason men have more iron in their body, especially in their blood, than women. So we think that's the reason men have higher cancer rate than women. We'll get to that in a bit. And also meat eater versus a vegan, or actually just the vegetarians, the cancer rate is 3 for meat eaters to 2 to 1 for vegetarians. Okay, so when chronic inflammation meet with iron, this reaction, this inorganic reaction will take place for sure, as long as the concentrations of each is high enough. So it's a 2 charged iron plus H2O2. This is from inflammation, and that will lead to a 3 charged iron. And this leads to hydroxide. This is a basic molecule, and hydroxyl radical. This is the strongest free radical our body can produce. Okay, so if this reaction only takes place once, it's not a big issue. But then if there are reducing elements in the nearby, then if that can reduce the 3 charges of iron back to 2 charges of iron, this reaction will continue. We did some statistical analysis on TCGA data. We found that for all cancers, they all use superoxide, as I mentioned earlier, as the reducing element, which is produced also by immune cells. So I wanted to make one point here, that there have been some large clinical studies that show that vitamin C is bad for cancer patients. And there are a couple of papers on this. I think that the observation is absolutely correct, but I personally think the the explanation may not be quite correct. We think the reason is that vitamin C can help to reduce 3 charged ion back to 2 charged ion. This will accelerate the Fenton reaction. We'll see the key things. So for this talk, I'll be focused on the alkaline molecule hydroxide. Okay, so another thing I wanted to give you some general idea about some strange behaviors of cancer. So we know that the cancer persistently divide, but in addition to that, there are many other highly conserved behaviors across different cancers, but people still do not have a general understanding why that is the case. So for example, about 100 years ago, there's a brilliant German scientist, Otto Warburg. He observed that between the 2 energy metabolisms our cells have, cancer cells prefer to use the less efficient, more ancient a process called the fermentation pathway or glycolysis pathway to produce ATP compared to the normal, more efficient pathway, something called the respiration pathway or the electron transport chain. So that's an observation, but there are still ongoing debates why that is the case. So we'll throw in our explanation in this talk. And even though people still do not quite understand why cancer cells have the Warburg effect, but this has been widely used because Warburg effect will directly lead to increase the consumption of cancer cells. Hence, that leads to increase the glucose metabolism, which is the basis for PET-CT detection for cancer, just to find whether someone's organ has a highly elevated glucose metabolism. So that is the observation, kind of strange behavior of cancer. Another interesting observation about cancer is synthesized DNA. They generally rely on de novo synthesis of DNA just by itself, rather than a cell can uptake the variation of this nucleotide. We can just assume it is a nucleotide. But so by uptaking it, it's going to be much more efficient, much more, it will be energetically, it will be more economic. So, but the cancers prefer to use de novo synthesis to synthesize the nucleotides for its DNA synthesis. We'll come back to this point later in the talk. So here's another kind of a strange behavior. So we know that if you remember amino acid metabolism, at the end of the metabolism, the cell will release the waste, which is ammonia. Our normal cells use this process called the urea cycle to produce and release ammonia. But interestingly, all cancer cells, based on our study, inhibit a urea cycle. So it's interesting. It's still, you can do amino acid metabolism, but it will not use this cycle, but using a very alternative, very long, very seemingly expensive process to release ammonia. So we'll not get into further details about this. So here's something probably many people do not know is this. So this is the curve of a cancer occurrence rate versus age. So basically this is age, and this is the frequency percentage of the frequency of people having this disease. So this is the plot for testicular cancer, which has a very early peak. It's about the third year of age of 30. So some other cancers also have a young peak age. So then you have to ask, what happens beyond this age? Why the cancer risk gets very high, then decline over age? Because quite often people think aging is a risk factor for cancer, but we found in the SEER report that virtually, probably except one or just few, very small number of cancers, virtually all cancers has this type of distribution. This is uniformular distribution, or not uniformular, unimodule distribution, but except that the peak may have a different age. So that's something we'll get to. Why this kind of a very strange behavior, even though some have been noticed, but not have been systematically studied. So we'll take all these things into consideration, come up with an explanation. Okay, so here's something probably not a lot of people study, but as we study cancer biology, we realize that this is something we have to study to understand some of the strange behaviors. So what the cellular polarity is, essentially you can think that is the infrastructure to support certain behavior, certain functions of a cell. So basically, without getting into the detail, you can think polarity is just the infrastructure for different functions. It requires a different polarity. So human cells have, when it's doing normal work, it has certain type of polarity there. When it's a device, it has other cell polarity. So basically, cell polarity defines what cells can do and also defines what the cell cannot do. So generally, it's the direct functions of cell polarity is related to anything related to orientation, movement, localization, large molecular assembly, that sort of thing. So we'll touch on this issue because it plays an important role in our analysis. So the key point is, so if the human cell has a human cell's polarity, this polarity will inhibit human cells to do things microbial cells do. Okay, so just remember that, then we'll come back to it. Okay, so basically that the more complex organisms tend to have a more sophisticated cell polarity. Simpler organisms will have a simpler cell polarity. You will see how during the cancer evolution, how the cell transformed the cell from the very complex cell to the very simple cell. We'll get to that later on. Okay, so now I want to use this example from bacterial biology. I think this is really the important the guiding principle in modern biology. It's kind of in spirit is consistent with Darwin's theory of evolution. So the idea of this, the story is very simple. So basically, this group of researchers, they put this bacteria, Pseudomonas, into the same environment with this phage that eats bacteria, phage 2. Okay, so they basically put them into the same environment. Some of like the bacteria may eat some type of the food that they eat. The phage lives on the bacteria. So after a while, they reach an equilibrium. Let's just assume that 50% in the environment is phage, 50% is Pseudomonas. Okay, so then, so they watched this for a long time. There's no fundamental changes in terms of, they're mainly interested in genomic, the genome of both. So what then they did one simple mutation on the bacteria, making it's difficult to be eaten or to be killed by phage 2. So then we see just making so that changes the life and the death rates. So that will making it more difficult to be killed. So the Pseudomonas population will grow and the phage 2 population start to shrink. So there's a group of researchers just closely monitoring these the genomes. Then once they reach a certain point, the phage 2 population becomes fairly small. Then suddenly, then they observed that these organisms started to grow back. And at the same time, they found that there were substantial mutations take place. Okay, only in phage 2, not in Pseudomonas. So then they reversed the experiment. They did one mutation in phage 2, making its attacking ability stronger. Then they found that the Pseudomonas population will shrink and the other population will grow. And at this time, the same thing happened. The Pseudomonas population will grow back after a while. And then at the same time, the researchers found a lot of mutations take place. So basically, this is there's a new terminology called the stress-induced mutagenesis, and which has been widely used in bacteria research and also in plant biology, based on my literature review. Okay, so basically, it's the main point here is it is really the stress that leads to mutation. Actually, people even know how those mutations, how the mutation takes place, because the stress increase the production of something called ROS, reactive oxygen species. That will lead to the increased mutation rate. Then some of them will be selected by the organism, which can be used to help them to overcome or adapt to the stress. Okay, so that's kind of the general principle. So we ask if the same principle applies to cancer, what are the main stressors and so on. Okay, so another thing, since we are talking about a new way of looking at the cancer, I shall say something about the previous work, the kind of the major work in the past. So over 150 years ago, there's a German physician, and also he does a lot of things. But basically, he proposed that it's the persistent irritation that is the main cause for cancer. To me, that's probably the most closest to some of the most current understanding about cancer. So we also mentioned the Warburg effect, that basically in 1960s, before the death of Warburg, he just basically said, not only cancer cells have this switched from respiration to fermentation, he also suggested this is the reason. I think he said, this is the sole reason for cancer development. Okay, so then we all know that the kind of ongoing thinking since about the 1970s, people believe it's the genomic mutations. The mutations is due to replication errors. When cell divides, some errors may be introduced. That is the basic cause of cancer. Okay, so that's basically my quick kind of a summary about what we need. That's kind of a prior knowledge we need to talk about the idea of my seminar here. So here is some general statistics I want to people know. It's even though we have been fighting cancer in a systematic way for almost like 5 decades, a lot of money put into this, and then we know that if you look at this curve, this is the death rate per person. Actually, this is normalized with respect to age, the longevity and the life expectancy. So we can see that the death rate is for women, it's about, you can see it's dropped somewhat, but you can see it's not that significant. Men, I think also you can see that how it changes over the years from 1930s to 2016. And based on another data collection, both are on the same history data. I do not know why they look a little bit different. So the top part is the cancer occurrence rate in the past 20 years. And the death rate, you can see this is the blue one is the death rate. Some people think it's pretty stable. So that's kind of the state of the art, regardless lots of once in a while we hear there's a revolution that can cure cancer and this and that, but some of these are not necessarily all supported by data. So we also know that for men, every 2 men, one of them will have cancer during his lifetime. For women, the rate is 1 out of 3 women will have cancer during her lifetime. This is not a worldwide statistics, and this data has been the statistics have been quite stable. Okay, so basically that we know these, and there are a lot of theories, there are a lot of ideas about why cancer takes place, why it's so difficult to treat, why it becomes drug resistant, all these things. And then all these kind of different, multiple generations of new technologies. And as I mentioned earlier, then the reality, is there. We know that the occurrence rate is still high. The death rate dropped somewhat, but still very high. And people still think cancer is a deadly disease and a very, very scary disease. I personally know few people who died young in their 30s, 40s. And so it's still a very, very, very scary story. Okay, so now we want to think about, can we think about, based on other research, can we say something? Have we learned any lessons? So here is based on my own literature review. So I study cancer by looking at the large quantities of data, tens of thousands of tissue samples. So come to a kind of a general understanding. To study cancer, you really have to treat the problem as essential as an ecosystem problem. It's not just the cancer cell. You have to look at the immune cells. You have to look at the other cells. You have to look at all the neural cells. All these things are involved in the development of cancer. So, but in the past, so basically these are just like people study ecosystem, not only study the organisms in the environment, they also need to look at all the basic nutrients, like where the carbon come from, where the phosphate come from, where all these other nutrients come from. So it's because of these things you have to really, really consider all of them to really start to have some understanding. But in the past, that because of the experimental limitation and we have largely focused on a fairly focused view rather than look at the bigger thing, because that's just reality when you study cancer cells in a culture plate, because that's all the things you can do because it's this experimental limitation. But now the good thing is with all the data available, because these are real tissue data at the different stages, in pre-cancerous stage and the post-cancerous stage, meaning that the metastatic cancer stage, now you can really see the whole history. So that's where we come in. So as I mentioned, we have all this data. We think probably the data have all the information we need, but we need to have a framework to learn how to extract the data and help us to develop a hypothesis and validate them. So that's the kind of the starting point. So before I'll tell you what we think is the approach we took. That I wanted to kind of basically say something about the stresses of the cancer cells are under in general. So basically these data are based on TCGA data. So most of our information are not from literature, are really directed from cancer tissue data. So we know the following: cancer cells generally consume a lot of more, 20, 30, even more nutrients, especially glucose, because of the reduced glucose to energy conversion rate. So they consume a lot of more nutrients, and the vast majority of them will die very quickly. Over 96, 97% of them die very quickly. So, but then they're under all kinds of pressures, stresses. They're under hypoxic, lack of oxygen stress. They're under oxidative stress. Then we will focus on pH-related stress today. So they're under all different stresses, but they still divide. So after a while, not just initially we come to this hypothesis, but after a while, we come up with this hypothesis. We think cancer cell division is a way to survive. because if they do not do it, the consequence will be even worse. So we propose, we hypothesis that if they do not divide, they will die 100%. Okay, so we have been using this guiding hypothesis to guide us to do our knowledge discovery. Okay, so we look at the cancer from the particular perspective, probably not the same as many people who focused on genomics aspect. We focused on metabolisms. So in case for people who are not very familiar with metabolism, essentially it's about what the cell eat, then convert them into energy, convert them into various biomolecules, then release metabolic wastes, like we talked about earlier, like urea, like ammonia. So here's a very simple metabolic process. Everybody should be able to understand. It's a CO2 plus water plus solar energy. Then by using the energy to change the CO2 together to form our sugar, that's what the plants essentially is all about. Then also as a side product, it produces oxygen. Okay, so we will study metabolism in cancer, but we will not study the normal metabolism of cancer. We study the altered metabolisms of cancer. These are, there's a word for that. It's called the metabolic reprogramming. They just, it refers to not the normal way of doing the same thing. Generally, these things are not pre-programmed. So quite often, these altered metabolisms, they are not induced by normal regulatory system. Usually they are regulated through epigenomic level activity. Basically, when dealing with stress, something just get activated. So we'll not get into further discussion about this. So here's the example. It's like this. In cancer, we often see this. The normal metabolism will go from A to B, and which is probably energetically optimal over many years of evolution. But the cancer find a new way to change that metabolism. Often it will consume more energy, but we think that at the end, the benefit is they can survive. Okay, so we'll look at a few examples, then we try to come up with a conclusion why that is the case. So here's one example, serine synthesis. So well, in cancer have elevated amino acid metabolism, particularly nucleotide metabolism. It needs to synthesize nucleotides. So cancer cells will synthesize serine, even though it can be, this serine can be uptaken from circulation, but cancer prefers to synthesize them inside the cell. And also our own work discovered that if the cancer is more malignant, it will have a higher level of reliance on serine synthesis. So then we wanted to use this as kind of an entry point to understand why. So we studied the detailed chemistry reactions and to understand why this is the case. So when we write down, not from the beginning, not from the glucose, but from like a glutamate, then we found that we write down the chemical reactions. So we studied this for a while. At the end, we thought maybe there's, because we do not really see anything special. So we thought maybe it's the proton that's special compared to uptaking serine through just some transporters. So that's our first kind of observation. Maybe that's the first time we thought it may have something to do with pH. So we think, okay, maybe it has something to do with acidification. At that time, we did not really know. But then we started to look at other metabolism. So we know that for all the cancers we studied, half of them will do something very, very strange. They will synthesize triglyceride, then immediately it will degrade it. So this is just so strange. So cancer cells not only just do this, essentially you do synthesis degradation. Cancer cells does this for multiple situations. Like cancer cells does glycolysis and also the reverse process, something called the gluconeogenesis. So this is represent a range of similar cases that we want to understand what's happening. So this is the molecular structure of triglyceride. These are the three fatty acids. So we focus on this part. This is the synthesis part of triglyceride. We see that there is this cycle. It produces, it's a big part of the synthesis process. We found that in this cycle, every time it goes through the cycle, the cell will produce a proton, and at the same time it will consume the CTP. CTP is kind of like similar to ATP. It's an energy. It's an energy currency or carrier. So the interesting thing is, in 1970s, people already found. Some people reported that some cancers overproduce CPTs, but at that time, there was no explanation, just the report. We think that probably they are used, the CTPs are being used to help to synthesize triglyceride, so it can produce a proton. So this is the other side of the triglyceride. For its degradation, we found its degradation also produce protons. So basically here you look, so basically every time when the cell cut one fatty acid, remember that's three tails, every time it cut a fatty acid, it will produce a proton. So for all three fatty acids removed, it will produce 3 protons. So we get, this is another example. Maybe we think maybe this is the general trend. Okay, so Warburg effect. So people have been talking about the Warburg effect for almost 100 years. So basically this shows in normal cells use this long reaction chain. Basically this is glycolysis followed by something called the TCA cycle, followed by a respiration chain. It will produce 30 some ATPs per glucose. But the cancer relies more on this simpler energy metabolism. It produces only 2 ATPs, so it's less efficient. So that will make cancer cells eat more glucose. So then we also study the chemistry of Warburg effect. Basically, by going through these reactions, we come to the conclusion that the ATPs produced by Warburg effect or by the glycolysis pathway, it generates one net protein. I mean, not one net proton. While ATP used, or while the ATP produced by the normal respiration chain, then when it's consumed, the overall thing is pH neutral. So basically, this is another example that the altered metabolism has something, seems to be that it likes to produce more protons. Okay, so this is the kind of the key part that's really related to why cancer cells continuously divide, and also cancer cells, they synthesize nucleotides and they use those nucleotides to synthesize DNA. Okay, so I hope people remember that there are 2 types of nucleotides. One is called the purine, one is called the pyrimidine. And also people reported by multiple authors that cancer tends to produce more purine than pyrimidines. There are various proposals, but there's no general consensus why they do it this way. So again, we use the same very simple-minded approach. It was, okay, let's look at the pH. So we studied the whole, it's not true. We'll go from the beginning, follow the whole de novo synthesis pathway to look at the overall chemical reaction. Basically, here we come up with for each purine, like it will produce an additional 8 proton when the purine is A. It will produce 9 proton when the purine is G. Similarly, oops, I'm sorry, this is overly sensitive. So then when it produces a pyrimidine, it produces 3 or 5 protons. Remember what I said earlier, researchers have reported that cancer cells tend to overproduce purine than pyrimidine. So we thought maybe, here purine to produce the purine to produce 8 or 9, to produce a pyrimidine to produce 3 or 5. Maybe proton production could be the driver here. So that's something we start to have the hypothesis. So basically, overall, we analyzed the 50 reprogrammed metabolism widely reported in the literature across 14 cancer types. It's the reason we did not cover all 20, 30, 32 types of cancer in TCGA is that these 14 has the most amount of data. That's only data limited. Some of them are missing stage 1, stage 2, or stage 4 tissues. So these are the 14 types that has most amount of data. So we found for each of the 50 some reprogrammed metabolisms, every one of them produce more protons compared to the original metabolism. This just really, really surprised us because for the following reason. Because we know, as we talked about earlier in the very beginning, we know the pH of cancer is higher than normal cells. But then you have all these metabolisms, so you can imagine each purine will produce 8 or 9 protons. Imagine you're getting the whole DNA synthesized. We're talking about the billions of protons in one cell. So you have all these protons being produced, but then the pH still go up. This is kind of counterintuitive. So based on this, we come to the hypothesis that there must be something secretly, we just don't know, that have been secretly, continuously produce hydroxide or something equivalent. Okay, so then remember we talked about chronic inflammation. Actually, the story is when we search for this, then we focused on this thing, something called the Fenton reaction. Remember, this is something we talked about. It's the result of a chronic inflammation and the iron overload in the local site. This will lead to persistent production of hydroxide. And there, remember, because of these two are the things that get consumed, these two are both produced by immune cells. And now iron is not consumed anymore. So it serves as a catalysis. Okay, so basically we think maybe this one continuously puts out hydroxide. It's possible the protons and the hydroxide, they're meant to be neutralize each other. Okay, so we did some simple kinetic-based analysis, something called the Michaelis-Menten kinetics. So people who took chemistry should be familiar with that. Actually, I'm teaching that right now. So basically, we look at these 5 elements and hydroxide concentration, H2O2 concentration, superoxide concentration, and iron, all these things. So these concentrations, the quantity, if without Fenton reaction, these quantities have no relationship. So we did quite a bit of analysis. We established a model. This model just, we almost like mathematical, we showed these things will be statistically related if and only if there is Fenton reaction. So this gives a tool to help us to probe cancer cells using statistical techniques to ask whether cancer cells have Fenton reaction. Even though people have reported this, want to really make sure all the cancer cells and the cancer tissues we study have Fenton reaction. So basically, based on this analysis, we found for all cancer tissues, their cytosol, mitochondria, extracellular matrix, and the cell surface, they all have Fenton reactions at a different level. And the focus of today's presentation will be on cytosol Fenton reaction. These other things I will not talk about because it involves some other stories. Okay, so we focused on cytosolic Fenton reactions. So we know all these things with we provide a lot of statistical evidence to show, yes, we think that they have Fenton reactions. In addition to that, we estimated that the rate of Fenton reaction can overwhelm the pH buffer very quickly based on the data. So we can do that. Other data already published. So now we want to compare the Fenton reaction continuously produce hydroxide, and all these altered metabolisms they produce protons. Do they use the to neutralize each other? So we did some relatively simple kinetics-based analysis to show essentially they produce these two things roughly about at the same scale. We cannot say they equal to each other because our model is not that accurate. Just we say they're at the same scale and they are correlated. Okay, so based on that, we predict that all the programmed metabolisms are induced to produce protons to keep pH stable. Remember, we said the pH has to be stable. If they're not stable, they will die. Okay, so now that's good news. The intracellular pH is stable, but then some real bad news now happens. Okay, so here's the kind of to summarize this part, and so this is it starts with chronic inflammation with iron accumulation to produce hydroxides. Initially, it can use some protons to keep the pH stable, but I said this one is not sustainable. So then it triggers all kind of triggered all kind of reprogrammed metabolisms. For this talk, I'm focusing on nucleotide synthesis, and I will touch on another things called sialic acids. We also did some work. These things collectively, they produce a lot of protons. I want to say all cancers have this reprogrammed metabolism to produce nucleotide synthesis. And some cancers may not have every single reprogrammed metabolism among the 50 we studied. But these three listed here, they're all conserved. They're conserved across all cancers, but at the different levels. Okay, so from here, we have to ask a question. In order to have the nucleotide synthesis as a way to acidify the cell, the nucleotide must have a way to be consumed or to be released. But this is the first challenge, which is very difficult. The reason is, as I said earlier, nucleotide is negatively charged. So you cannot simply just release them in large quantities because that would change the neutrality of the cell. So cell must have some other ways to get rid of the nucleotides. So then we ask, is it possible that cell division is a way to get rid of the nucleotides which are being rapidly synthesized in order to neutralize the protons? So basically here you can think that if we inhibit nucleotide synthesis, this is the main acidifier, then it cannot produce enough protons, the cell will die. So this is the stress I was looking for, but is this the really one? Now we want to produce some evidence. So even though this seems to be very, very probably impossible, but this is all unicellular organisms do, because all unicellular, based on my study, literature review. So we look at the yeast, we look at the E. coli, we look at a whole bunch of other things, more complex things, and then basically they all basically keep eating. Then first they will produce ATP. When the ATP level is too high, they will switch to nucleotide synthesis. When nucleotide concentration is high enough, they couple it with sugar and use the nucleotide sugar or a variation of nucleotide as the cue for cell proliferation. Okay, so is it possible somehow this mechanism is still somewhere hidden in our genome and being reactivated by the cancer cells? So, okay, so that's kind of the question, is that possible? So and also want to mention one thing. There have been the theory about something called the cancer stem cells theory. So, but there is a difference. We should know that cancer cells division is a symmetric cell division, meaning that the cell will divide, then both daughters can further divide. But the human stem cell, except for the very, very earliest few generations, all the other stem cells will divide, produce another stem cell under the differentiated cell or functional cell. So this will not work because we think this when the cell stop dividing, the cell will die. So basically, this model will not work. And we also know cancer cells use symmetric cell division. Okay, so basically, we need to understand whether the human cells may have somehow become like a unicell organism. Okay, so basically, we systematically studied all the mutations in all the genomes of the 14 cancer types. Overall, 7,000 some genomes. We studied all the mutations. We found something just shocking. 50%. So traditionally, people think only a few of the mutations are relevant to cancer. They give them a name called the driver mutations. But we just decided to study all the mutations. We found 50% of all the genomic mutations of cancer are related to simplification of the cell polarity. Remember, we said the cell polarity is the infrastructure for cell. So we found the vast majority of them, they all related to cell polarity. But then I wanted to point out that many of these cell polarity genes in the past are called the tumor suppressor genes, like a P53, like a PIK3CA, like APC. These things, they are all part of the cell polarity machinery. But then if you keep those cell polarity, cancer cannot take place. I think probably for that reason, they are giving the name tumor suppressor genes. Okay, so then in addition, I think I got this wrong. In addition, people found, this is not our work, people found that most of the oncogenes, they're all related to nutrients, like RAS, like mTOR, like all these, many of these genes, they all relate. So if you look at what RAS does in yeast, it's actually all these things related to report of availability of amino acids or sugar or something. Okay, so basically based on this, we have a manuscript in writing. The paper essentially is there. So basically in this paper, we predict cancer cells are highly simplified cells. Essentially, they behave like a unicellular cell. And the evidence mainly comes from the cell polarity. Basically, we mapped most of the cancer genes, and to this organism, I will not reveal the name yet. We will soon. That we mapped, I don't remember, most of the cancer genes, active genes, to this particular organism, and the mutations largely mapped to human cell polarity genes, and the oncogene mutations are mapped to ancient nutrient-related genes. Okay, so this is our kind of the main prediction for this talk and also for this manuscript. So here's the overall model. We start from here, then we set the mutation, the protons, the metabolic reprogramming, and they keep pH stable. Then there are some other side products in order to produce protons. Nucleotides, they have a place to go, and we predict that this is done through this transformation, very fundamental transformation through systematic mutations, and it enabled this essentially converted that into a unicellular cell. It enabled these cells to do symmetric cell division. Then also, I want to mention another thing. Sialic acid is another major acidifier. And so sialic acid also has another interesting property. It is generally employed on the surface of the cell, like on the surface of the cell, as the capping molecule of glycans. Okay, so another interesting thing is all the sialic acid, each sialic acid carries the proton. No, carries the electron. So this will make these cells have an increasingly higher level accumulation of electrons. So this will lead to cell-cell repulsion. So recently there is a paper published, not recently, about almost 10 years ago, someone studied that when you put mechanical pressure on cells, this will lead to cell migration. So by combining our data and his result, we proposed a model how sialic acid production can drive cell migration and metastasis. So we're also doing something. So basically, I will use the next model. So then we have this prediction that based on our data, which essentially says it's the distinct combination of different metabolic reprograms that gives rise to various cancerous behaviors. So using this, we are currently doing some other further analysis. Right now, we are building a cancer cell cycle model. Interestingly, up to this point, people have been assuming that cancer cells essentially using the normal human cell cycle, but our data suggests otherwise. So we are building a new cancer cell model. We're also building a cell polarity model, as I mentioned in the previous slide. We have this paper in preparation. So then we also do some mouse work through collaboration with a group in China, and in the same time, right now we are doing some drug resistant issues. This also related to another type of metabolic reprogramming, and we are writing up the paper on metastatic cancer. We found that they are driven by different drivers. Okay, so there are some other things we are doing. Essentially, that's my whole talk. And this is basically this model. Using this model, we can explain various things. And so, for example, we can predict. We know what dictates the rate of cancer cell division, what dictates the potential of cancer metastasis, and why the age-dependent cancer occurrence rate has this unimodule distribution and some other issues. So why we are writing up another paper on why different organs have a different cancer occurrence rates. So for example, colon has fairly high cancer occurrence rates, but the small intestine has a very low cancer occurrence rate. Why is that? We find has something basic in the chemistry of the organ that plays key roles. So basically, that one of the purpose of the talk is to hopefully we can recruit, not recruit, we seek more collaborations. Right now I have a small lab. We have a lot of things we wanted to do, but not able to do. So we are looking for people to work with. But then we wrote in a manuscript as an invited prospective paper, essentially summarizing the work here. So if you're interested, you can contact me. As the last slides, here's the list of the people who contributed to work, and except the few people who are still working in the lab, virtually all of them, including multiple people from IOB, they all become professors somewhere. Okay, I'll stop here. Thank you.
All right. Thank you, Ying. Thanks for the great seminar. So I want to just open this up for questions for Ying, if you have.
Yeah.
So Ying, if I may just get started. So again, thanks for the great seminar. I was just wondering if many of your observations related to sort of the alterations in metabolic profiles also hold when you look at single cell sort of sequencing data. Because I was wondering, to what extent the heterogeneity, the TCGA data can contribute.
Oh, that's a very good question. Actually, yeah, that's something we have been thinking about. But since I have a small lab, I personally do not do a single cell based data analysis. So I'm collaborating with the former student, Xia Chao, who graduated from here a few years ago, now assistant professor at the Indiana University. We are collaborating to answer exactly the question you are asking, see whether for places, if the Ross-Fenton reaction, do they have the same level of metabolic changes? I think that's a great question.
Okay,
we hope by actually that involves a new master student, also from IOB, recommended to me by Jan Mrazek, and she is actually conducting that research. We hope that we'll get some results by the summer.
Okay.
Hey, Dr. Xu, great presentation.
Thank you.
So I have a question. It's more of a general question. I can't really get into the biochemistry as much, but in the beginning of the talk, you mentioned the correlation between red meat and colon cancer. You also talked a lot about the Warburg effect, which is actually brought up by a lot of proponents of ketogenic diets as sort of a support for eating more meat. So I wonder if you think, or if you've seen any papers that if you include, because I think on that, so this was like one of your first slides, like the countries, the graph of the countries.
Yeah.
If you look at actually that same graph probably has a very similar relationship with refined sugar intake. And it's probably a combination of the two, considering the Warburg effect, which, like I said, proponents of the ketogenic diet say that you can actually starve the cancer of the sugar that it needs by just eating a fully meat-based diet.
That's an interesting thought, but I'm not sure whether it's related, because we know the following, that if you look at the cancer metabolism, you essentially have to convert everything to glucose. So that means if you eat amino acid-based food, it will add burden to cancer cells, because it cannot directly use, it's hard for them to use amino acid-based nutrient. You have to convert essentially amino acid back to glucose. Then you do that. Essentially, you do things like gluconeogenesis. So that, I think that's the reason. I think if that's the case, I would think just the glucose-based food is easy for cancer to utilize, and the amino acid-based food will add additional burden to cancer cells. So that's my thing. So for this one, I think it's probably mostly it's iron, it's beef, because we get most of our iron from beef, and this is very consistent. If you look at the people who are low, like anemic patients, like if you look at the sickle cell disease patients, their cancer rate versus a healthy population is also 1 to 3. So I think there are just a lot of such data that support the idea of iron plays an important role. But sugar could also too. But so that's
Yeah.
Go ahead.
Well, I mean, I'll keep talking if I'll let someone else ask a question.
There was Mike, if you don't mind. So there is a chat box. There's a question by Kelvin. I just wanted to read it out. So with regard to cancer stem cells, could stem cells be closer to the unicellular-like phenotype and thus the ultimate source of most cancers?
So here's just basically, as I mentioned earlier, I thought I really, because I do not have that kind of data. So, but from my understanding about the cancer stem cell, so here's what I know. Very fortunate, I have a postdoc in China who has a strong background in developmental biology. So she told me that just only for human cells, only the very first few generations, they do symmetric cell division. Then for any specific organs like liver stem cell, they all do asymmetric cell division. So that means that, so for any asymmetric cell division, it will not adequate for cell to survive, because essentially everything, the new cell divide, the cell will be killed. So then you always end up with one cell, or because the cells do not divide based on our model, that will be killed. So based on that, I think cancer stem cell may not be adequate, but that's just my very, very personal view.
Okay. Any other questions for Dr. Xu?
Yeah, I have one.
Okay, yeah.
Hi, Ying.
Hi.
It was really cool. I'll need probably a couple of days to process all that information.
You can have the PPT.
I have. This may be a naive question because I don't know much about the human physiology and cancer, but when you mentioned the relationship of cancer and fermentation, I was thinking like muscle cells use fermentation during exercise, right? So is there any relationship between occurrence of cancer and exercise, like long distance runners? Are they more likely to have cancer?
Very good one. Actually, very interesting. Muscle cells have something, it releases, I think it's IL-6. IL-6 is when you exercise, muscle cells release that. That's anti-inflammation. So I think that's the reason this is very difficult for muscle cells to develop cancer. Some people say, it's because the muscle cells do not divide. I think that may not necessarily be the case, because under stress, we know now, based on just the general knowledge, any cell can do reverse de-differentiation, and they become cells that can divide. So I think that's probably the big reason.
Okay, so
exercise is good.
Okay, so I'm gonna think about it and maybe start exercising. All right. Thank you.
All right, any other questions for Ying?
Anyone from students?
So real quick, if no one else wants to.
Mike is a student, yeah. Mike,
Yes,
take it away.
So back to your point about iron. So the rate of cancer in men is higher than the rate of colon cancer in women.
Yeah.
But yet, women consume and require more iron. Are you saying that women have less iron?
No, yes, of course. Women have less iron, that's for sure. That's known, established. Because as I mentioned, iron, when they enter into our body, they will not leave unless we lose blood.
Right. But the thing is, what you're seeing is just localized iron concentration, not necessarily
still,
systemic iron concentrations, right? So it suggests that there's a problem with just a specific area rather than a systemic problem, right? So
that's a very good point.
To me, that doesn't really point to red meat at all.
Okay. So here's this. Okay. So for example, that one person's baseline is this high, another person's iron baseline is this high. So it will be easier for this person to reach the threshold to have a Fenton reaction. It will be easier. Then in terms of localized iron overload, that actually has something to do with chronic inflammation. This one is already established by the tissue repair community. People found if there's a place have a high level of those superoxide or hydrogen peroxide, they can oxidize. If the concentration is high enough, they can oxidize red blood cells. So the red blood cells does not have a membrane repair mechanism. So if they are highly oxidized, they will die. Essentially, they will be swallowed by macrophages. The macrophages will spit out the iron. Then it will be sequestered by the local cells. So that leads to the local. It's really the result of chronic inflammation. So don't have chronic inflammation.
Good point. Thank you.
Okay, if there are no more questions, I think in the interest of time, let's all thank Ying. Well, thank you, Ying. And if you need to reach Dr. Xu, please email him directly and follow up on the seminar.
Thank you.
Okay.
May I ask a question?
Yes.
Oh, okay. All right.
So I'm wondering, so have Professor, your lab or your collaborator tried to do cell level or like animal models to test this iron-driven cancer development?
Okay, that's a very good question. Yes, we do some cell. We do mouse work through a fairly extensive collaboration in a group in China, and actually that we already got the data generated a while back. We're still just busy, reduce the capacity, just all these things. We have not been able to finish the analysis, but I think the data strongly support our model. So that mouse work is basically, we feed the mouse with certain carcinogens that lead to liver cancer. These mouse they have full and healthy immune systems. So yes, so we do that, and iron gets accumulated in the very early stage of cancer. I mean, the development even before cancer takes place. Then also regarding iron, the relationship between iron and the cancer, there have been published studies that have shown that if you feed mouse with high iron food, that will lead to cancer. I think if you double like the normal iron level, like just double that in the food, that will lead to cancer development in mouse.
Okay. Thank you very much.
All right. Thank you so much, Ying.
Thank you.
Thank you for the, yeah. Okay.
Bye-bye.
Bye-bye. Thank you.